Hello and welcome everyone to today's presentation titled Non-Viral Large-Scale Delivery and Gene Editing Tools for T-Cell Manufacturing. This webinar is part of the second annual event in the Cell and Gene Therapy Virtual Event Series. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Lonza. For more information about Lonza, please go to www.lonza.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of our presentation. If you have trouble seeing or hearing our presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I'd like to introduce today's speakers, Andrea Toell, Associate Director, Portfolio Management at Lonza, and Stefanie Muethel, Team Lead R&D at Lonza. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Andrea, you may now begin your presentation. Thank you, Andrea and Stefanie, for your informative presentation. We will now begin the live Q&A portion of our webinar. If you have any questions that you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen, and we will answer as many questions as we have time for. So let's get started. Our first question is, how did you establish the optimal conditions for the CRISPR tools?
With trial and error, I would say. So initially, we established the procedure in small scale, and first we tested several guide RNAs to find the one giving the best knockout. And then we tested several HDRTs with different lengths and provided them in different modes like plasmid, nanoplasmid, or double-stranded DNA. And in the end, we used a synthetic guide RNA, SPCas9, and double-stranded DNA from GenScript. And for optimal ratios and concentrations, we tested or used what was already published and found our optimal condition based on that. And as you may have seen during the presentation, this is different for RAP11A and TRAC, so you may need to optimize it for your specific locus. So we did a lot of optimization of the general conditions in small scale before we transferred the setup to large scale.
Perfect. Thank you so much. Our next question, if programs are not directly transferable from small scale, how does the scale up work?
Mm. It works in a way that we will provide a protocol with specific recommendations, which programs to try for a certain cell cargo combination. And it's typically a set of 3, maybe 4, sometimes 5 programs that can be then tested in the 500 microliter fixed volume format to save cells and cargo. And then the best program selected can then be transferred to either larger volumes in the fixed volume format or also to the flow-through format.
Oh, thank you so much for that. Our next question, what is the minimum volume that can be processed in the flow-through format?
Technically, you could go down to 1 milliliter. However, the ratio between cells lost in the flow path due to cells sticking to surfaces or transfected cells collected or principally cells lost and transfected cells that have been collected may be suboptimal. So currently we recommend not to go below 3 milliliter. However, we are also looking into that more closely now to get a better idea about the potential loss and what is exactly lost, whether it's viable transfected cells, untransfected cells, or cell debris. And depending on what it is, and if it's viable transfected cells, we will look into options how to recover these in order to be able to even work with lower volumes if needed.
Thank you, Andrea. We have one more question that has come through. Is the LV, oh, apologies, it looks like we have one more that's come in addition to that. So again, I'll just give a reminder, if anyone has questions, please submit through the Ask a Question button. The Ask a Question panel, and then submit at the bottom. We'll get through as many of these as we have time for. Our next question, is the LV unit nucleofection already in use in clinical trials?
Yes. So the first generation LV unit is in about 10 early clinical trials which are ongoing, and one late clinical trial has started. And there are several projects in preclinical phases. And for sure, with the new LV Unit Pro, nothing has started yet because we just launched it.
Right. Thank you for that. Our next question, how does the efficiency of nucleic acid delivery by the 4D nucleofector vary among different human primary cell types? Example, immune cells under hospital lab condition.
I mean, if you look at different cell types, I mean, they are differently susceptible to transfection. So some are harder to transfect, and some are easier to transfect. And especially immune cells probably belong to the more tricky to transfect ones. So you have to figure out the optimal conditions for each and any cell type, and for some cell type, like for example for B cells or so, maximum transfection efficiencies you can achieve are around, I don't know, 30% for plasmid DNA, let's say, whereas with T cells you may get up to 60, 70%, depending a little bit whether it's activated or resting T cells. And what you always have to consider if you work with immune cells is that you have some donor dependencies. And so you may see donor variabilities from experiment to experiment in a certain range, and that could affect efficiency as well as viability. So you always have to consider that if you think about robustness of the process. So yeah.
Great. Thank you so much for that response. It looks like that closes our Q&A portion of our presentation. I'd like to again thank Andrea and Stefanie for your time today and your important research. Before we go, I would like to thank our audience for joining us today, and also our sponsor, Lonza, for sponsoring today's webinar. Please do visit the Lonza booth in the exhibit hall after our webinar for more information and resources. Any questions submitted during the on-demand period will be addressed via the speaker addressed by the speaker via the contact information that was provided at the time of registration. This webcast can be viewed on demand for 1 year until October 15th, 2026, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until then, take care, everyone, and have a great rest of your day. Bye-bye.
Bye. Thank you.